Hello everyone. Today we are going to see about skeletal muscle contraction. So here we will be dealing about the A band, H band and all the different bands as well as a short overview about how the contraction happens. So this is the structure of the sarcomere. So this is a muscle fiber and here we have numerous myofibrils are shown and this is the basic functional unit, that is the sarcomere. If you see here in the sarcomere we have the dark pink lines. So these dark pink lines are the myosin which is denoted in this way. But whereas the light lines which are in between all these myosin, they are the actin, so which are present over here. Now the middle line, so that is this line in the middle, it is the M line. So I used to remember that the line which is present in the middle is the M line, and the entire myosin which we see, that is the entire pink, that is the A band. But whereas that A band is the entire myosin which is also overlapped by actin. But there is a part of the myosin which is not overlapped by actin, and that is known as the H band. So in first aid, a clue was given for this A band, that is A always stays the constant. So even during contraction, A band is the one which doesn't increase or decrease. So the A stands for A, that is always remaining constant. So this is the one, and this is the I band where the two actin, this is the actin over here, like say for example here, and the actin over the other side. So the actin which is not overlapped by myosin is the I band. So this is the sarcomere structure. So let me just brief you again. So this is the A band and the middle line is the M line, and this is the H band, that is a myosin which is not overlapped by actin is the H band, and the I band is the actin. So now this part where there is the actin on either side, it is connected by this line. It looks like a Z, isn't it? So this line is known as the Z line. So let's see in this diagram. So as I told you, the darker band is the A band, which is the myosin. And whereas if you see in the middle, there is a line which is the M line or abbreviated for middle. And in between there is a lighter color also, so which is the H band. Now here if we see, here we do have the actin, this is the I band. And from one end to the other end is the Z to Z, that is the sarcomere. How does muscle contraction happen? Muscle contraction happens when the actin and the myosin overlap. So during this time, the actin and the myosin come together. Therefore, the H zone and the I band will shorten. But the A band is a length which will remain unchanged. So let's see how this contraction happens. So initially what happens is that action potential will open the presynaptic. So even before that, so the mind or the brain will think like it has to contract a part of the muscle. So once that thought has been provoked, then the thought is sent via the nerve membrane such that the nerve will meet with the muscle. That area is known as the motor end plate. So when it comes to the motor end plate, when that action potential or when that impulse is near the motor end plate, it will open the presynaptic voltage gated calcium channels, including the acetylcholine release. Once the synapse, so I can help you. So this is the this is the nerve, this is the end part of the nerve. So once there is a presynaptic voltage gated calcium channels are present. So this is the presynaptic, right? So here the calcium channels are present. So once the calcium enters inside, then from inside the nerve we have acetylcholine which is released. Once the acetylcholine is released, it will lead to the muscle cell depolarization over here. So this is a muscle cell, this area is undergoing depolarization, and this area is the motor end plate. So once the acetylcholine is released, it will result in muscle cell depolarization. This depolarization will travel over the entire, so it will go in this direction, it will go in this direction over the entire muscle cell, and it will also dive deep into the muscles via the T tubules. So these are the T tubules. These are the T tubules. The muscle, this depolarization will enter into these T tubules. Once the depolarization enters into the T tubules, it will induce conformational change at the voltage sensitive dihydropyridine receptor. So here we have a receptor. So this is the dihydropyridine receptor, and that dihydropyridine receptor is mechanically coupled with ryanodine receptors. So once the ryanodine receptor is also activated, now this is the yellow part is a sarcoplasmic reticulum. The calcium is released from the sarcoplasmic reticulum into the cytoplasm. Once the calcium is released into the cytoplasm, this is the actin myosin. This is where the actin myosin is present. So we had already seen that this is the actin and this is the myosin. This is the myosin and this is the actin. Once the calcium is released into the cytoplasm, the calcium. So let's elaborate this area where this is the actin filament and this is the myosin filament. Once calcium is released into this area, then the calcium will bind to the troponin. Why should it bind to the troponin? So this is the troponin. Why should it bind to the troponin? It binds to the troponin because there is something known as tropomyosin, as you see here. This tropomyosin will mask the area where the myosin is supposed to attach to the actin, thereby causing the contraction. Only if the tropomyosin moves away, that area where the myosin is supposed to get attached with the actin can be exposed. For the tropomyosin, for this blue area to move away from the myosin binding site, we need troponin to move this tropomyosin away. For the tropomyosin to cause this action, it needs calcium. So hence, when the calcium binds to troponin, that will cause a shifting of the tropomyosin to expose the myosin binding site. Now the myosin head will bind strongly to the actin. This myosin head will bind strongly to the actin, forming a cross bridge. Phosphate is then released, initiating the power stroke. So during the power stroke, force is produced as the myosin pulls on this thin filament, resulting in the muscle shortening. So with the muscle shortening, it will also result in shortening of H as well as I bands and between the Z lines. The A band will remain the same. A band is always the same length. So this is how the muscle contraction happens. So we have the action potential, and after that just an overview of this. So there is an action potential. After that we have calcium influx. So once calcium enters, there is acetylcholine release. Once acetylcholine is released, there is depolarization and more calcium is released, resulting in tropomyosin being moved away and the myosin getting attached with actin, and thereby the muscle contraction happens. This is how the cross bridge cycle happens, where there is first myosin will bind, then there is a power stroke. After that it will get detached and ATP hydrolysis. So for this cross bridge formation, we need a power stroke, we need detachment and ATP hydrolysis to happen. Now let's see about the muscle fibers. There are two important muscle fibers. One is a slow twitch muscle fiber and other is the fast twitch muscle fiber. How should one remember what is a slow twitch or a type 1 fiber? So this is a muscle fiber where it is mostly useful for marathon runners. So if we see the marathon runners, that muscle, logically if we think, that muscle should not get fatigue easily, isn't it? Otherwise they cannot run the marathon. So these muscles will be slow fatigue. So now it has to withstand a longer distance. In that case, it needs to be aerobic and it needs to have a lot of mitochondria to produce its own fuel, and which is, since there is many mitochondria present, it is also red in color, but it does not have glycogen because it does not need glycogen for this type of exercise. Therefore, the slow twitch muscle fiber will be red in color, and it is the one which is predominantly present in marathon runners. On the other hand, there is a fast twitch muscle fiber which is mostly present in sprint runners. So therefore they will be able to run quickly. So they will be they will be very quick. So for that to happen, it needs to have a high energy source. Therefore these muscles will have high glycogen. So it need not need to have an myoglobin. It need not have to utilize an aerobic, because it has a high glycogen content, and therefore with that glycogen they can cross over that small distance in a very high speed. So therefore it will be anaerobic, and it will be white in color, and there will be less aerobic capacity because there are only fewer mitochondria. With this we come to the end of the slide. So I have one question to tell you, which has been asked in NBME exams. So it is like a 28-year-old male who has a femoral nerve injury resulting in denervation of rectus femoris. So rectus femoris is supplied by femoral nerve. So once there's a femoral nerve injury, it has undergone a denervation. So question asks, what is most likely to be seen during nerve recovery? During nerve recovery, there is fiber type grouping of type 1 and type 2 muscle cells. You need to know that reinnervation of muscle results in aberrant so once a muscle is getting reinnervated, it will result in aberrant reorganization of type 1 and type 2 muscle units. This aberrant reorganization is called as the fiber type grouping. That's it. And there is also a step 1 offline form where they ask why ATP does not fall appreciably during a series of muscle twitches. So ideally it is ATP is supposed to fall appreciably, isn't it? Why doesn't it form? This is because ATP is quickly regenerating from creatinine phosphate. Because of the quick regeneration from creatinine phosphate, it does not fall appreciably. With this, we come to the end of the presentation. Thank you for being with me all this while. We have officially finished our topic. So, but with this, I have an announcement to make. I am starting my mentoring lessons for USMLE graduates, USMLE aspirants, and medical graduates. If you like my video and if you like my slides, and if you want to get mentored by me, you can go ahead and send a message in the comments. Looking forward to see you succeed. Have a good day.